So again, you need to understand four main ways of destroying your protein's function or DNA is kind of similar way, and its structure as well. So denaturation is one way of doing that, enzymatic degradation is another, exposure to oxidation or aggregation. Aggregation is what? Your proteins lose their structure, and then when they come together randomly, they will stick together through random hydrophobic interaction. That's the aggregation. So functional protein become garbage that are all stuck together. That's aggregation. So these are the four main ways. Understand? There may be an exam question on this. Okay. And the question is, how do you protect your protein or other API molecules from losing their structure and function? Okay, by providing the lab solution that has these features. Okay, so your lab solution should have buffer salt, which will resist the pH change. Okay, so that's why you need to have buffer in the biological solution. Okay, also your solution should have certain amount of salt because your bodily fluid has some salt in there as well. So your biological molecule is adapted to that environment. So too much salt or too little salt is not good because all those ionic bonds will be broken up. So ionic strength. So add some salt in your in your solution. So about what? 0.15 to 2 molar solution is good. Okay, and the cofactors are what? Enzymes normally need cofactors because they need like metal ions and things to maintain the structure and activity. So you may need to add cofactors, or you may need to deprive cofactors if you want to protect your proteins from enzymatic degradation. Okay, so cofactors are important. And detergents, let's say you have protein that is strongly hydrophobic, so it tends to be aggregated, right? Then by keeping your protein in this detergent solution, your protein will be protected from aggregation because detergents are what? These are like your dishwashing soap that has amphivalent structure. Amphivalent structure means what? One side of the detergent has long hydrocarbon chain like lipid and fat. The other side it has a charged characteristic like SO minus and whatever the charge characteristics. So by providing that long hydrophobic tail, it can bind with your protein that is also hydrophobic through hydrophobic interaction. And by wrapping your protein around, and it'll make it dissolved in the buffer that may be hydrophilic because it has that charged group. So detergent will protect your protein from degradation. So examples are like SDS or Triton X100 are good examples of the detergent. So your biological solutions will contain a lot of these factors, okay, when they protect your proteins or APIs from damages on their structures. Okay. Another way, artificial denaturants. Okay, so you can add certain amount of SDS, okay, or some other denaturants like guanidine salt, TCA, ethanol, acetone, methanol, chloroform, or ammonium sulfate and PEG to make your protein denatured a little bit, not completely. Okay, they may be helpful to maintain their integrity so certain under certain conditions. Okay, and then reducing agents, like I said, the cysteine to cysteine disulfide bonds can be broken on if they are exposed to very reducing condition. Okay, well, those disulfide bonds are formed under kind of oxidative condition. So these chemicals like dithiothranitol or beta mercaptol ethanol is a strong reducing agents, okay, because they have a lot of SHs in them. So by adding these guys in your biological solution, you can protect the integrity of disulfide bonds. Okay, they're all important. And then metals and chelating agents. Like I said, metals are important cofactors for the enzyme. So chelators are what? Those are the chemicals that can bind with metal very efficiently. So if you want to suck out the metals from enzymes, so enzymes will be inactivated, you add these chelating agents. Like EDTA is a good example. EDTA can bind with metal ions, okay, efficiently. By doing so, it can inactivate the enzyme that can chew up your DNA or protein, okay. And the antibacterial agents. So any kind of solution, if you keep the solution a long time in the room temperature or fridge, wherever, there's a tendency that there may be some bioburden or bacterial cells can grow in that solution. Okay, to prevent that from happening, you need to add, preservatives like sodium azide and things like that. Those are added to the solution, then they will kill any bioburden, the bacterial cells, so they will not grow in your solution. And sometimes protease inhibitors can be added. These are all important protease inhibitors, which will bind and inactivate the protease enzymes, so your protein will be protected from them. And temperature is also important. When you work with your proteins, you normally work under kind of cold condition, like 4 degrees in Celsius. That's about your fridge temperature, okay? Because under that condition, that temperature, what enzymes are inactivated very slow. There's a less tendency that your protein will be degraded by the enzyme if you keep your protein in there. Okay, and absorption. Absorption means what? Your protein can stick to its container wherever it is stored or dissolved. It may randomly stick on the glass, the material on your container or plastic material. Then when they stick and they don't come off, then you will lose your protein. So it's not good. So, absorption could be a problem. So you may want to, add some anti-absorbing chemicals to protect your protein from there. Okay. So not only the proteins are your API, sometimes DNA, RNA, nucleic acid could be your API as well. For example, like vaccines that are RNA based, in that case, RNA molecule is really important. Let's think about nucleic acid. DNA, RNA, they are deoxyribonucleic acid or ribonucleic acid. They have these bases in them, okay, CTU, A, B. So those are pyrimidine, 3 different kinds, purine, 2 different kinds. And so these are bases making up DNA, RNA, okay? And then there are 2 different sugar molecules, ribosugar or deoxyribosugar. And then there's a phosphate stuck in it. So if you combine all 3, phosphate, ribosugar, and base, that's a nucleotide. That's a building block of DNA or RNA. Okay, if you put those building blocks together, that is a nucleic acid, either DNA or RNA. The difference is, do you use this deoxyribosugar there? Then it's going to be a DNA. Do you use ribosugar there? Then that's going to be an RNA molecule. One of the important different thing is RNA has uracil in them, but DNA has thymine in them. Okay, so which base it has, that also determines the integrity of DNA or RNA as well. So anyway, DNA molecules exist as in double-stranded structure. So one strand here, one other strand there. Between those, there are what? Hydrogen bonds between their bases, like a double bond between A and T, and triple bonds between G and C and C and G. Okay, so that's how those 2 strands come together and stay together. That's the structure of the DNA typically. And these are hydrogen bonds, which are non-covalent bonds. Those bonds could be easily broken up. Okay. So under what condition those bonds could be broken up? Denaturing condition, like the protein case. So if you expose your DNA to, acidic condition or basic condition, yes, those hydrogen bonds will be broken up. And then if you expose your DNA to a high temperature condition, also hydrogen bonds will be broken up. High salt, same way. So those are all denaturing conditions that will compromise the structure of your DNA. Okay. And then degradation could also happen, just like proteins. There are enzymes that can chew up your DNA, RNA molecule. We call those enzymes nuclease enzymes. They can break down the bonds that are formed between nucleotides. What do you call those? Phosphodiester bonds. Those are very strong covalent bonds, and those bonds could be broken up by these enzymes, nuclease enzymes. So we call those degradation. Okay, and shearing of chromosomal DNA by physical force. During your DNA RNA isolation, you may use centrifugation, you may use sonication, you may use multiple pipetting, and those physical forces could also damage your DNA or RNA as well. So those are all denaturing or damaging conditions as well. So there are solutions that will prevent the degradation or denaturation of DNA, RNA. Of course, by adding buffer, you can keep the pH constant. Ionic strength, again, you need to keep some salt in there in your solution. Organic solvent will remove any protein-based enzymes that can chew up your DNA, RNA. Nuclease enzymes will be inactivated by those. Precipitation, same way, your DNA, RNA could be coming down as a pellet if you add some salt with the presence of strong ethanol and isopropanol. And then you wash your pellet away and dissolve in a tea buffer again. That's a way of cleaning up your DNA, RNA molecules. Nuclease to selectively remove RNA. So there are RNA only chewing enzymes, DNA only chewing enzymes. Depends on what molecule you use. You can use alternative enzyme that can remove the molecules that you don't want. And nuclease inhibitors as well, protease K or EDTA, can inhibit the nuclease activity. So these are all important tips when you try to maintain the structure with DNA RNA when you deal with them. Okay. Now, not only you will have to deal with the biological molecules, sometimes you will have to deal with the cells, right? So you will grow a bunch of CHO cells to produce API, or sometimes you will have to grow a bunch of fungi, yeast cells, or bacterial cells like E. coli cells, because those are genetically modified cells that can express the genes that are code for the API molecules, right? Then how do you keep those cells intact and happy so they will continuously grow, divide, and make more APIs for you, right? So you need to understand the condition where these cells are happy, right? So just like biological molecules, cells are made up with a bunch of those biological molecules in them. So pretty much the similar conditions. The only difference is cells are independent. They'll do their own metabolism to get their energy. So you have to feed them with the energy source such as sugar. They will break down the sugar through cellular respiration process and capture energy, and then they'll use those energy to maintain the structure and divide, go through cell division to make more daughter cells, right? So those are the conditions that are important, right? So of course, in the solution that will keep your cells, you need to provide certain amount of salt. Okay, sodium chloride. You're probably familiar with these terms: hypertonic solution, hypotonic solution, or isotonic solution, right? Remember those terms? Isotonic, or hypotonic solution, or hypertonic solution. If you expose a living cell under any of those three conditions, what's going to happen? So hyper means what? It's a high salt solution compared to how much salt your cell has. And if the solution outside has more salt, what's going to happen? There's going to be what? Osmosis, movement of water. Water will move from where to where? It's high concentration to low concentration. Relatively, inside a cell, if it's dipped in a solution that has a lot of salt, outside there will be less water in there, within a given space. So water will move from inside a cell to outside cell. When that happens, cells will shrink and they may die because of it, right? And in opposite case, if cells are dissolved in the solution that has very little salt, what's going to happen? The water will move from outside to inside. So your cells will expand and eventually it's going to burst and die, right? So the best condition where your cell should be what? Isotonic condition. That's the same salt concentration. Then it's not going to lose any water. So it's going to maintain its structure, right? So you need to figure out what that solution is. About 0.9% sodium chloride. That will be the saline solution. When you have, dehydration, you get that saline needle injection. So that's what it is, okay? So cells will grow or maintain in a solution called growth medium, because that growth medium has the perfect pH adjusted with the buffer. And then, like I said, it'll have some sugar. So the cells can feed on those sugar to capture energy. Essential salts. Salts are important, like I said. And the vitamins are also needed, trace elements, micronutrients, hormones, antibiotics sometimes to kill other contaminants. So those are the main ingredients of cell growth media, where you're going to grow your cells and keep your cells happy, right? So if you're growing bacterial cells, okay, prokaryotic cells, what are the things that you need? Okay, minimal media is made up with certain conditions. Okay, it may have a complex medium, not purified. It's like a dead body of yeast. That's yeast extract because it has a bunch of sugar, protein, all the rich nutrients in there. Sometimes could be blood of certain animals, and that'll be the good rich source of nutrients that your cells needs, because bacterial cells are what? Those are the decomposers. They used to live on the dead body, right? So they'll be fine with those dead bodies, right? And then selective media, if you want to grow on your cells, nothing else, you need to provide certain antibiotics on there. And differential media, besides the antibiotics, you can add different types of media to maintain the integrity of your cell culture. Okay. The main ingredients of bacterial growth media will contain a lot of water. Of course, the growth medium is water-based. Sometimes you need agar to make it solid, okay, to make a pretty full of medium. And other nutrients like complex medium like yeast, dead bodies of yeast, yeast extract, peptones, all those are hydrolyzed milk, a rich source of proteins. And the tryptones digest casein, the milk protein. Those are good source of nutrients, right? Source of energy, sugar need to provide, of course. And then sodium, potassium, phosphate, those are the buffer salt, okay, you need to provide. Trace element, growth factors, supplements, preparing, there's an instruction on how to prepare bacterial media in your textbook. Take a look at those. Examples are LB broth and LB agar, okay?